This is your cell and molecular biology course, and I'm your course instructor Vinya Tandul. Welcome to this session on DNA repair mechanisms and their significance. So today we will explore how cells protect their genetic information and why these repair processes are crucial for health and survival. So let's begin with introduction to DNA damage. Every day, the DNA in our cells faces damage from various sources. These include environmental factors like UV radiation, chemicals, and even normal cellular processes that can create errors. If this damage goes unrepaired, it can lead to mutations, cancer, and genetic diseases. So, why is DNA repair essential? DNA repair is the body's defense system against genetic errors. It preserves the integrity of the genome and prevents mutations from accumulating and is essential for preventing diseases like cancer. So, without efficient DNA repair, cells would quickly lose their function and also their viability. So, this is why DNA repair is essential. And there are types of DNA damage. Let's first understand the types of damage that can occur. So common forms include single strand breaks, double strand breaks, base mismatches, and chemical modifications to the bases. Each type of damage requires a specific repair mechanism to correct it. So these are the commonly occurring types of DNA damage. Now we will see how the DNA repair mechanisms for each of these occurs. Base excision repair, or you can say BER. The first mechanism is base excision repair. BER fixes small non-helix distorting damage such as the deamination or oxidation of bases. So this basis is nothing but the nitrogenous bases which we have studied earlier in the structure of DNA. So special enzymes called glycosylases recognize the faulty base, whichever is damaged, and it recognizes the faulty base, and then it will cut it out and replace it with the correct one. So this will keep the DNA code accurate. So this is how base excision repair occurs. The other type of repair is nucleotide excision repair, or you can say NER. So this pathway handles bulky lesions like timing dimers caused by UV light. So there is a team of proteins that would cut out a short segment of the damaged strand, and then the DNA polymerase fills it. That is, it will fill the gap with correct nucleotides. So this is how nucleotide excision repair occurs. So due to UV radiation, a certain part of the DNA segment is damaged, and then a special type of proteins, which is the group of proteins which will identify this damaged region, and then it will remove it. And then this is, as usual, is replaced by the DNA polymerase enzyme by the nucleotides, which should be currently present, which should be correctly present in that particular area. So the other type is the mismatch repair, or you can say MMR. So mismatch repair corrects errors that escape proofreading during DNA replication. So this repair occurs during the DNA replication if any nucleotide is mismatched with the newly formed DNA which is replicated. It detects the mismatched bases like when adenine pairs with cytosine instead of thymine, and then it will remove the incorrect segment. So this reduces mutation rates and is vital for genome stability. So this is how mismatch repair or MMR takes place. So in the illustration, you can see the pair which is mismatched, and then a sequence of DNA which is mismatched. It is being recognized, and then the nucleotides are being removed, and then it is replaced so that the genetic code is maintained. So other type of repairs include HR and NHEJ. So double-strand breaks are the most dangerous form of DNA damage. So two main repair systems fix them. One is homologous recombination, or you can say HR, which uses a sister chromatid as a template for accurate repair. So this is the HR. And the other one, that is the other type of mechanism, is the non-homologous end joining, or NHEJ, which directly joins broken ends but can be error prone. So these are the two types of repair mechanism which occurs during the double strand break, which is very dangerous uh, form of DNA damage. So one is homologous recombination and the other one is non-homologous end joining, but it is prone to error. So these are some of the mechanisms through which the DNA repair occurs. So now let's see significance of DNA repair mechanisms. These repair mechanism pathways are essential not just for survival but also for preventing diseases. The defects in DNA repair genes can lead to conditions like xeroderma pigmentosum, and the other condition is Lynch syndrome and even breast cancer. So thus, we can say that DNA repair helps maintain health and prevent genetic disorders. So this is the significance of DNA repair mechanisms. So DNA repair in cancer therapy. Interestingly, some cancer therapies target DNA repair mechanisms. They use drugs like PARP inhibitors, which block the repair in cancer cells, causing them to die. So this will prevent the repair mechanisms in the cancer cell so that the cancer cells would die. This approach is effective against tumors with existing repair defects such as BRCA mutated cancers, which is uh, for breast cancer actually. So this is how DNA repair is used in cancer therapy, and we can consider it in case of aging. Over time, DNA repair efficiency declines, leading to an accumulation of mutations, and this contributes to aging and age-related diseases. Enhancing the repair pathways is an essential, is an area of interest for promoting longevity and healthy aging. So, DNA repair mechanisms are vital guardians of our genetic information, and they correct errors and they protect against disease, while they also ensure the faithful transmission of DNA during the cell division. So, understanding these processes not only explains how life maintains its code, but also opens door to new therapies and interventions. Thank you for joining me in this exploration of DNA mechanisms. I hope this session has deepened your understanding of the cellular strategies that keep our DNA safe and also our bodies healthy. So this is about DNA repair mechanisms. See you in the next video.